Good morning. My name is Sabine Franke. I'm a toxicologic veterinary expert regulatory review pathologist at CFSAN FDA. I will be talking about carcinogenicity testing at CFSAN FDA, FDA needs, context of use, the decision framework surrounding carcinogenicity. I am speaking as a subject matter expert. My opinions should not be construed as FDA policy or the position of the agency. I have no conflict of interest to declare. A little bit of background. FDA is part of the Department of Health and Human Services. FDA is the Food and Drug Administration. Specifically, CFSAN is the Center for Food Safety and Applied Nutrition, and we will be talking about the regulated products that are being dealt with in the Office of Food Additive Safety within CFSAN. The organization I already mentioned, and the organization is depicted here in more detail. Working under the commissioner of the FDA are many centers and regulatory offices. The Center for Food Safety and Applied Nutrition is located here. However, it should be noted that we are scheduled for a reorganization for the Human Foods Program. There's a link that explains this future activity in more detail. The mission of the FDA is to ensure the public health pertaining to its many regulated products. CFSAN specifically promotes and ensures the protection of the public health as it pertains to the national food supply, its safety, sanitary, wholesome, and honest labeling. We also currently host cosmetics and cosmetic products. OFAS, the Office of Food Safety and Applied Nutrition, is responsible for the protecting of the consumer's health, ensuring the safety of food ingredients. Those are substances that are added to food and food contact materials. The jurisdiction: we regulate food chemicals, food additives, color additives, food contact substances, food constituents, contaminants, impurities, as well as substances that are generally recognized as safe. There are several laws that govern the work of CFSAN FDA. The 1938 Food, Drug, and Cosmetic Act, the 1958 Food Additive Amendment, the 1960 Color Additive Amendments, the 1996 Food Quality Protection Act, and the 1997 FDA Modernization Act. In order to understand the regulating background that lead to carcinogenicity testing within the safety assessment of CFSAN FDA, I would like to run some concepts by you that are important in order to understand it better. The safety standard informs the toxicologic testing recommendations. And the 1958 Food Additive Amendment is the foundation for the pre-market approval of food additives, which also include food contact substances. There's a general safety clause in Title 21, the USC paragraph 348C, that provides that food additives must be demonstrated to be safe under its condition of intended use before it can be added to food. Specifically, it is stated with regard to safety that safety requires proof of reasonable certainty that no harm will result from the intended use of the additive. Congress noted at the time that it is unattainable to aim for no harm, so that the zero risk is not attainable. The resulting safety standard is therefore reasonable certainty of no harm. We also need to understand the Delaney clause for food additives. It is found in Title 21 USC paragraph 348 C38. It starts with a general safety clause. No additive should be added to food if it fails to establish that under the conditions of its use, it is safe. And then there is a provision that speaks specifically to cancer. the additive cannot be deemed safe if it induces cancer when ingested by man or animals, or if it is found after tests which are appropriate for the evaluation of the safety of the food additive and induces cancer in man or animals. We will come back to this particular wording a little bit later in this talk. So where does the Delaney clause apply to the materials that we regulate? It pertains to food additives. It pertains to color additives. It also pertains to food contact substances. It does not pertain to brass substances. There is a specific exception. And it doesn't pertain to constituents, contaminants, and impurities. Those are regulated under the constituent policy with a quantitative risk assessment. Next to the law, there are many guidances out there and information that the FDA provides in order for anyone who wants to interact with the agency on the regulated products. For example, should there be a interest in a food additive petition, here is the information out on the internet. It is quite informative, ranging from an overview to specific administrative information to expertise such as chemistry, toxicology, microbial information that is needed. Similarly, there is information on how to put together a notification for food contact substances, as well as notifications that pertain to the GRAS notification program. To get a little more specific, when it comes to testing safety within CFSAN, one needs to look at the testing battery and how it is informed. Specifically, the chemical structure and the exposure, which is the cumulative exposure of humans, inform a specific concern level that is determined by the agency. The concern level translates to the minimum set of recommended toxicity tests. This is a different depiction of this information. I know it's quite small and you won't be able to read some of the details, but it is put here more for illustration. So if you see a concern level of low, then the testing requirement is qualitatively and quantitatively low. Only a few tests are required, such as genetic toxicity testing, as well as short-term toxicity tests. Should the concern level be high, then the number of required tests improves, as well as its complexity. That type of testing requirement also includes the carcinogenicity studies in rodents, of which we will talk about a little more later. In the context of testing studies that are submitted to the agency, we have to realize that we are asking for quality standards for all materials that support safety assessments of food ingredients. These quality standards are, for example, the Good Laboratory Practice Regulation or guidance, such as the FDA Red Book guidance. The FDA Reg Book came out in 1982 and has since been updated multiple times. It is also referred to as the toxicological principles for the safety assessment of food ingredients. Other materials that help understand the quality standards that we are looking for are published in the general literature. I'm giving here an example for the quality standards pertaining to pathology data. Pathology data are very important for carcinogenicity assessments. This article was written by my mentor and colleague, Dr. Prem Dua, and his colleague in 1988. Yes, this article is old, but general quality standards still apply. I copied the table number 2 out of this article. And here we highlight that we are still today asked for, are the animals healthy? How is the diagnostic terminology applied? Is it harmonized with global standards? Is it correct? Is it informative? And then the narrative. is required to be informative and interpretive. So here is an example of information pertaining to carcinogenicity testing. One of those tests is the 2-year bioassay in rodents. FDA requests an in utero phase. You can find this information in the aforementioned Red Book guidance. However, FDA promotes also the development of alternative test methods to support the replacement, the reduction, and the refinement of animal testing. Specifically, the TOX21 effort at CFSAN and FDA helps the modernization of toxicological approaches to safety testing. And this is a logical requirement as scientific progress demands it. We apply various non-animal tools for the prioritization and the weight of evidence building on a case-by-case basis to the assessment of safety of food ingredients. For example, we utilize predictive modeling, QSAR. We also use read-across or genetic toxicity testing. In the subnote of this slide, you find several links to other activities that CFSAN FDA engages in order to foster the non-animal testing alternatives. In this context, I also would like to bring to your attention that there is a long history of CFSAN FDA engaging in this type of work. This is an excerpt out of the draft of the 1993 Red Book 2, where the agency actually requests and requires for alternatives to whole animal modeling. Particularly here, alternatives to whole animal vertebrate experimentation. It is important, though, that we encourage and recommend that the agency is being consulted for any type of testing before the submitting party engages in testing. What types of data does CFSAN FDA see with regard to safety packages? As outlined before, data are specific to the substance. It's a case-by-case profile. As I mentioned before, the testing battery is depending on structure and human exposure. Therefore, the safety assessment is a multidisciplinary team approach. It involves chemists, toxicologists, environmental scientists, microbiologists, biologists, and pathologists, as well as consumer safety officers. The specific data that we receive thereby are chemistry data, environmental data, genetic, toxicity, pathology data, as well as data that inform the risk assessment and calculations for the estimated daily intake level, the EDI, as well as the information that allows us to put together an acceptable daily intake level, the ADI. All of this goes in an administrative record for each specific petition. The way we use the information, again, going back to a case-by-case scenario, the assessment is is specific to the circumstances. So carcinogenicity data are only required when there is a high concern level, as I pointed out before. However, if there is information already available for carcinogenicity data, then they will also inform the safety assessment, even if the petitioner has not provided this information. So FDA conducts their own research. The carcinogenicity data inform the overall safety assessment. If the carcinogenicity assessment is negative, then the materials will be used for respective calculations in the risk assessment, as well as in the determinations of the EDI and the ADI. Is the determination, however, deemed positive by FDA, then the food additive cannot be approved because of the Delaney clause. And that decision is independent of exposure or mechanism. So now I walk you through how the carcinogenicity assessment is actually performed. Each and every carcinogenicity has their own professional scientists looking at specific sections of the material provided. The material is resource intensive and involves voluminous data sets and a cadre of subject matter experts. Should one of the expertises have a positive signal, let's say in the example provided below, the pathology report comes up saying, hey, there's actually evidence that there is potential carcinogenicity. Then a second layer of intensive scientific scrutiny and deliberation ensue. So depicted here, a positive signal in one of the subject matter experts report would trigger a center expert meeting that can be a cancer assessment committee meeting or other expert committee meetings that are furnished with many scientific expertises and the review of all the relevant data. Sometimes consultations of other disciplines are included, such as medical experts from CEDAR, and ultimately there will be a vote and the generation of a memorandum of carcinogenicity. This memorandum includes the deliberations, the reported and recorded discussions, and the results, and it can inform the resulting recommendations for regulatory action. So we were asked what type of data we need in order to assess human relevance. In this context, it's important to know that feeding studies, clinical feeding studies, epidemiological studies, or even mechanistic study for carcinogenicity pertaining to food safety are often not available to CFSAN. Therefore, we rely on animal study data and alternative testing data. In this context, also, the Delaney clause has to be mentioned again. I pointed out before that the ingestion of a food chemical under review that induces cancer in man or animals, or has been shown to do that in appropriate evaluations, cannot be approved. It should be noted here that the word animal appears twice right next to man. That means in the context of the Delaney clause, the food additive and food additive safety, an animal carcinogen is as relevant as a human carcinogen. If FDA has found a food additive to be a carcinogen in animals, then the human relevance discussion is generally irrelevant. We were also asked if there are needs that potential NAMs can be in assistance with. And we responded that we would agree to all of the below: compound screening, compound prioritization, hazard identification, potency evaluation, point of departure, risk assessment. We are looking for validated or validatable tools that support regulatory decision-making. I would also mention that NAM might stand for non-animal models or new alternative methods. Now I would like to introduce you to a quote that I found in a publication of one of my colleagues at CEDAR FDA, Todd Boissier and his colleagues. Had a publication in 2015. The overarching message is carcinogenicity testing is difficult to model. I modified this quote a little bit to carcinogenicity is one of the most difficult toxicological endpoints to predict, given its mechanistic complexity and high variability of long-term study data. Consequently, it is in particular important to understand the value and the limitations of all models. Again, there is an omission of a word in the original text, but I think it's important to apply this to all modeling before selecting the model for a specific analysis. The 2-year rodent cancer bioassay, of which we have heard a little bit already, is one of the many study types that inform carcinogenicity safety assessments, but it's usually at the forefront of its criticism of carcinogenicity testing. A must read for all those who might attempt efforts to replace it is this article by Bayer, Beck, and Lewandowski. It speaks specifically to the historical perspective of the use of animal bioassays and the prediction of carcinogenicity. The takeaway of this assay is that the 2-year rodent bioassay was originally developed to address urgent regulatory needs, specifically food safety testing addressing the Delaney clause. And as you have heard, the Delaney clause speaks not only to human, but more so as they are easily to attain animal carcinogens. This assay was originally designed to detect animal carcinogens, and the exploration to humans was much simpler. The only prudent course of action is to assume that chemicals which are carcinogenic in animals could also be so in man. The assay standardization and harmonization is essential for any tool that is perceived to be used. The bioassay in its inception was a wild jungle. It took years to get it to a point where it was functional, and that was achieved through standardization and harmonization. The 2-year bioassay has been increasingly complicated by add-on studies that are far beyond its intended purpose. A key difficulty in carcinogenicity is the interpretation. If interpretation between mammalian systems, which are rats and mice on one side and humans on the other, is uncertain, then how uncertain would that make extrapolations from proposed NAMs to human cancer risk? With this, I'm asking, before we let the animal model out this door, we have to address several uncertainties. These uncertainties are quantitative in nature. How many NAMs would it take to replace an entire whole animal system? 10? Maybe 10,000, maybe a minute. However, there are also qualitative questions that have to be addressed. So in order to have NAMs, they need to be producing reasonable certainty, predictivity, as well as protectivity for carcinogenicity assessments. With that, I thank you for your attention, and IEHS for putting the venue forth, my colleagues for valuable assistance with this talk, and for feedback on the preparation of this talk. Here are some references that you might find helpful, and I hope this information was helpful and informative. Thank you.